In this video we will discuss about this Siglec-10 and the Siglec-10 which is evidently expressed in the pancreatic cancer's tumor microenvironment. So what they are able to find out and how they come up to these conclusions. So what it is, they check the publicly available pancreatic cancer data or more specifically this tumor microenvironment of the pancreatic cancer data, and within that they check the single cell RNA-seq data, and they are able to see that the Siglec-10s are expressed in most of the myeloid cell subset populations that can be seen in this panel A, that about 21.8% macrophages has this are Siglec-10 positive, and even in this CD4 T plus cells are also Siglec-10 positive, dendritic cells are also Siglec-10 positives, MDSC is also Siglec-10 positives. So they think that since the Siglec-10 seems to be evidently overexpressed in this TME, this could be plays a role towards immunosuppression. So this is their hypothesis. First, they check the different compositions of this Siglec-10 positive populations in the tumor microenvironment, and they are able to see that around 60% of the Siglec-10 positive cells are macrophages. The rest of them are other different subsets of myeloid cells or the dendritic cells and monocytes. And when they compare it with the other Siglecs that are reported in the literatures to be overexpressed in the tumor microenvironment, such as Siglec-7, 9, 15, they see that Siglec-10s are usually more overexpressed compared to the other Siglecs. And afterwards, they wanted to check if there is any gene change expressions or change in gene expressions occurs in the Siglec-10 positives myeloid cells compared to its control. So for that, they check the cancer TGCA, that is the Cancer Genome Atlas, and from this they are able to find out that for the macrophages, the genes that are upregulated belongs to the immunosuppressions and also the proliferation and metastasis of the cells, such as TGF beta signaling pathways are upregulated. And similar things they have seen for the myeloid-derived suppressor cells also, that TGF beta signaling pathways are TGF beta receptor is upregulated, indicating that TGF beta signaling pathways upregulated, and that might cause more proliferation and metastatic characteristics. Afterwards, in the dendritic cells, what they are able to see that the antigen presentations receptors such as HLA-DOA and HLA-DOB are seems to be reduced because of the high expressions of these Siglec-10s. Though it seems to be like HLA-DOB is seems to be high, however, this expressions values are significantly lower compared to the Siglec-10 positive cells. Um, Compared to the Siglec-10 positive cells that are present in the tumor microenvironment. And afterwards, they check these Siglec-10 expressions in different stages of the cells or different stages of the cancer cells, such as the borderline or the adjacent cells has the lower expressions of the Siglec-10s, whereas the resectables and locally advanced cells has high expression of Siglec-10s, and even the metastatic cells also shows high expressions of the Siglec-10, indicating that Siglec-10s could also be the markers for the metastatic cell lines. But it is not restricted to the metastatic cells. The other stages um, uh, also contains the high Siglec-10 expressions. Afterwards, they check it in the whole tissues of the compare it with the normal tissues and this pancreatic adenocarcinoma tissues, and they are able to find that this pancreatic adenocarcinoma tissues has more Siglec-10 expressions compared to the normal tissues. And when they did it for the survival probabilities, when they compute the survival probability from this TGCA data, they are able to find out that high Siglec populations has or high Siglec patient has lower survival rate compared to the low Siglec patients. So, and after that, they finally come to the point that, or finally end this figure with a point that the Siglec-10 could be a Glyco-immuno-checkpoint. That is, it suppress the immune systems through its glycan. That is, the TME of the pancreatic cancer suppress the immune systems through its modified glycan expressions or overexpressed glycan expressions of Siglec-10. This is what they finally concluded from the results that are presented in this figure. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.